In this demo, we'll walk you through the correct use of a compound microscope. You'll need to be able to view histology slides throughout 2 semesters of PMB 2264, and you'll be much more successful at viewing the slides and learning the material if you know how to use the microscope correctly. Also, keep in mind that during the practical exams, one of the skills you'll be expected to demonstrate is the ability to view, focus, and identify histologic samples using a compound microscope. If you follow the simple steps in this demo, you can be confident that you'll be able to view the required slides and identify the cells, structures, and tissues on the slides. First, be sure your microscope is plugged in and turned on. You can adjust the light using the dial on the side of the base of the scope. The next thing that many students do is to grab a slide and put it on the stage. But that's not always the correct initial step. Take a moment and see if you can identify what's wrong with this initial setup to view a slide. What's wrong here is that a high-power objective is in place, and this is a relatively common error when using microscopes in the teaching lab. And you'll need these higher power objectives later on, but initially you should always view a slide using the 4x or the scanning objective. So before you even place the slide on the stage, be sure that the 4x scanning objective is in place. When you're viewing through the 4x objective, this will give you a wide field of view and a large working distance to focus the image. This simple tip will vastly improve your experience viewing histology specimens with the microscope. Only after the 4x objective is in place and the stage is racked down, you can place the slide onto the stage and be sure to secure it with the stage clip. Next, you'll use the coaxial stage controls to position the slide to be sure the light is illuminating the specimen. You can check this visually with the naked eye. There's no need to view the slide using the oculars just yet. Once the slide is in place, you're ready to view the slide and focus the image. The scopes we use in lab are binocular, which means they have 2 ocular lenses. And the best images can be seen if you look through the oculars using both eyes. If you need to, you can adjust the interpupillary distance by sliding the oculars. While you're viewing the slide under the 4x objective, you'll move the stage up using the coarse focus knob. And when the image is relatively sharp, you can use the fine focus knob to fine focus the image. Once you have the image in clear focus using the 4x objective, you can then rotate the nose piece so that the 10x objective is in place. You should always advance sequentially from the 4x objective to the 10x and so on. As you move to a higher power objective, you'll notice that the working distance, which is the distance between the slide and the lens, is decreased. From this point forward, you should only use the fine focus control. There are 2 reasons for this. First, when you're viewing under the 10x objective lens, the working distance between the lens and the slide is much shorter, and moving the stage using the coarse focus knob can result in accidentally breaking the slide or breaking the lens. Second, these scopes are parfocal. This means that when you switch objective lenses, the image should stay relatively in focus. If you start to use the coarse control, you'll quickly pull the image out of the plane of view, and it could be very difficult to refocus using the higher power objectives. Once you have focused and viewed the image under the 10x objective, if you want to view the image at a higher power, you can repeat and move up to the 40x objective. But remember, always focus and view the images using the objectives sequentially, 4x, then 10x, then 40x. The next higher power objective is the 100x objective, which is also called the oil immersion objective. It's very important to never use this objective on your scopes in the lab. Occasionally, the lab staff may set up a demo slide that requires oil immersion using the 100x objective. If this is the case, don't switch to another objective or the oil will be transferred to the other lenses. If you do need to clean the lens, please be sure to ask your instructor for special lens paper. Only this lens paper should be used to clean the lenses with, as any other paper product like a Kim wipe or a paper towel can scratch the lenses. When you finish viewing the slide, you should first move the stage all the way down, Then rotate the nose piece back to the 4x objective and remove the slide. When you're done for the day, be sure to turn off the light and unplug the microscope. Finally, if you need to transport the microscope, always remember to carry it with two hands, one under the base and one holding the scope by the neck. Keep these simple steps in mind when you're using the compound microscope. You'll be much more successful in focusing and viewing the histology slides in the upcoming labs. Number 1, always begin viewing and focusing using the 4x scanning objective lens. Number 2, focus the image under one objective lens before sequentially moving to the next higher power objective. Number 3, when viewing through the higher power objectives, only use the fine focus control and not the coarse focus control. Number 4, in this lab, avoid using the 100x oil immersion lens. Finally, if you're having any trouble focusing or viewing the slides with the microscope in the lab, be sure to ask your lab instructor for help. We hope this tutorial will be useful to you in the upcoming histology lab, as well as throughout the labs in this course.